I'm an assistant professor at Karolinska Institutet, working, my main line of research is now frailty. Also, we're studying various aspects of biological aging, but then this frailty is now the center of our work. And I'm not sure how familiar you are with frailty, if you're medical researchers working with elderly cohorts, or if some of you are aging researchers. So I'll try to walk you through all the concepts and potentially things that are not familiar to you. Let me see, can I go from here? Right. Okay, so first I will give a bit of background. What is frailty? How can one measure it? And how does frailty relate to other aging phenotypes? There are many rather similar aging diseases or phenotypes, so I will go through a bit of them. And then I will go through some rather methodological approaches. How can one assess causes and consequences of frailty? And then I will present some of our own studies into frailty. But first, a bit of the background. So conceptually, frailty is defined as an aging-associated decline in physiological reserves and function across multi-organ system, and this makes the individual vulnerable to adverse outcomes. And it's been known for several decades that frailty is strongly predictive of mortality, falls, fractures, a whole host of adverse outcomes. And it's not only so that frailty is a distinct phenotype on its own without any relevance to other outcomes. So this is pretty well known now also, that if a person has cancer or some other severe comorbidity, frailty among those persons can really exaggerate outcomes and treatment responses. So now in the clinics, at least here in Sweden, wherever possible, clinicians in pre-operative units, they make frailty assessments to decide, to make care decisions and to decide which kind of treatment the patient is able to go through and what is the expected outcome. And if a person is really frail or relatively high on a frailty scale, then some treatments are not, they are contraindicated, and they are not supported. And then it basically looks like this, that when a person is not frail, so managing well, then a relatively minor stressor, say urinary tract infection, does not cause any, after the infection is cured, does not cause any change in the person's functioning, and then the person returns back to the original level after the disease is gone. But then if a person is really frail, so then this relatively minor stressor can cause a disproportionate change in health status. And then even after this pathogen is cleared, return back to the normal level or original level is very unlikely. And this is where the negative outcomes, such as dependency and mortality and recurrent hospitalizations, happen. And then how can one measure frailty? So, well, there are currently more than 30 different scales, maybe even 40. Some suit better for research purposes. Some are mostly used in the clinic. Of course, there's always preferred choice based on the resources and time. And then this clinical frailty scale is the one that is, I could say, most often used in the clinic, and it's based on this 9-stage or score across 9 categories. And this assessment is made by a trained medical practitioner, usually a doctor or sometimes trained nurse. So this is assigned this level to a person, and then based on this, some treatment decisions or prognostics can be made. Then there's the frail scale. This is something I haven't used personally, other than unlike this. And then there is Fried phenotypic model, sometimes called FP. This is based on low walking speed, low grip strength, low physical activity, exhaustion, and involuntary weight loss. And I will tell you in a bit in exactly how this is assessed. And then in research, the Fried model is also used there, but then a really popular and well-validated model is the Rockwood Frailty Index, sometimes called deficit accumulation model, because it measures the accumulation of different deficits. And then this new one that we've also used, hospital frailty risk score, that is based solely on ICD codes. And then it's readily accessible in hospital records, in any record linkages where ICD codes are collected or available. And then this freed phenotypic model, so it's quite crude because based on these 5 criteria that I just mentioned, the individuals are classified into these 3 categories of non-frail. If the person has 0 of these criteria, 1 or 2 is pre-frail, and 3 or more classifies the individual as frail. So this is a rather crude, but at the same time very effective tool to stratify the risk in the individuals. And then it is the frailty index that I mentioned, and it measures the accumulation of deficits. And these deficits sounds a bit vague, but they can be signs, symptoms, diseases, difficulties in daily functioning, items on psychosocial well-being, and so on, as long as they meet sort of standard inclusion criteria. And the inclusion criteria are that they cannot be overly common in the population. So let's say in your study population, more than 80% shouldn't have. If you, for example, want to make poor vision or poor hearing as one of your items, so more than less than 20% in the population should not have it, so that it wouldn't be too common. Sometimes we say that some items that saturate too early cannot be used, that are too common. On the other hand, they cannot be too rare either. So the prevalence of each item that is to be included in this frailty index should be at least 1%. And they should cover a broad range of health domains. So this is by definition, this frailty index is multidimensional. So it should cover more than just medical aspects or functioning or psychosocial well-being. So all these areas need to be covered so that it works as it should. And then it also needs to have a minimum amount of these items. A good minimum amount is 30 or 35, I'd say. So then it doesn't really matter. The whole beauty of this frailty index is that it doesn't really matter what is the exact nature of the items, as long as you have many enough and as long as they cover the whole range of health domains. So then you can calculate or count, sum up the deficits that the individual has and divide by the total number of deficits considered, and then you arrive to a frailty index. That is a score that ranges from 1 to 0, from 0 to 1. The 1 is a rather theoretical limit, because it has been shown that in any population, regardless of what items are considered, nobody is usually able to tolerate greater than 0.7 level of this frailty index. So that is a peculiar fact of that whatever items you consider, 0.7 starts to be lethal. And then this has been shown that if you have, for example, had one cohort and then you assess 30 samples and then 30 other items, and you create 2 frailty indices based on 2 items in your cohort, as long as they meet the criteria, you basically arrive to the same level of frailty, regardless of the exact nature of this. So this is sometimes also called inherent vulnerability. So vulnerability is something that is that manifests regardless on how you actually capture it. So this was a bit of background on it. And then for these properties that it's continuous and it's really sensitive, it also works for younger individuals. And although frailty it is traditionally considered as a syndrome of the oldest old or old, but we've shown in our recent work that it actually matters for individuals at midlife too. So it's not just the oldest old. So of course, individuals who are, younger adults, they don't have these absolute high numbers of frailty, but then even small increases matter among them too. And this is just a list of example items, what can be included in the frailty index. This is 1/3 of the items that we have in one of our studies. So they can be diseases or activities of daily living, things like that. And then you score these and then you sum up the score and divide by the total number. Then sometimes people think it's quite easy to think that, okay, frailty, comorbidity, and disability, aren't they one and the same? And yes, there is a degree of overlap between disability, comorbidity, and frailty, but frailty is still a distinct geriatric syndrome or syndrome, and it can be defined without using any information on disability or comorbidity as well. And then frailty and cognitive decline, they are also very closely related phenotypes. And it has been even discussed, can these be separated? Are they so intertwined? One study is showing that they are really intertwined. They go hand in hand when observed longitudinally. This study also suggested that they might have a common pathological basis in the brain. In this case, it's the brain pathology that is driving both. So this is, of course, quite extreme case in individuals who already have Alzheimer's disease. But then again, there is another nice systematic review and meta-analysis showing that that in individuals who have cognitive decline but no dementia yet, there isn't that great of an overlap actually with frailty. So they are distinct phenotypes. Then, but since the order of incidence is always so that frailty tends to come first and dementia always later in later life. So predictions have been made so that frailty is used as an exposure to model the risk of dementia. And it has been tested for Alzheimer's disease, vascular dementia, and all dementia. So all these, well, all dementia and then these two most common forms of dementia. And here, here the assessment is made so that robust, so non-frail group is compared to frail versus pre-frail. So along the three categories in the Fried model. And then indeed, it seems that frailty is predictive of dementia, especially the individuals in the frail group have higher risk or higher odds of dementia. Most clearest relationship is seen with vascular dementia. And then another paper or systematic review looking at the different cognitive disorders was trying to disentangle, okay, which type of dementia is frailty most strongly related to. And then they found that it's a non-AD and vascular dementia where the greatest effect is. And then it's also been shown that that even though if frailty was a different entity than cognitive decline or dementia, it's been shown that the degree of frailty modifies the manifestations of Alzheimer's disease. So the higher your frailty level is, it somehow makes the brain pathology changes more pronounced. So it works as kind of in statistical terms as a moderator. But this is the same basically as with cancer. So any complication, any other problem you might have, they become more severe, more pronounced, the more frail you are. Right. And then a bit of the causes and consequences. These figures are just to illustrate how complex the whole scenario is, how hard it actually is to write like clear relationship DAGs or however you want to formulate them. So you want to pose your research questions really clearly and think about all these interconnected things so that you can actually answer the questions that you want, because frailty is in so many ways related to other outcomes. And that is actually the thing with why it is so good predictor, or let's say sensitive predictor of many aging outcomes. It is because it is so involved in so many processes, but at the same time it is also very unspecific because it's involved in so many conditions. So highly specific, highly sensitive, but relatively unspecific. And if you want to disentangle these relationships, the best ways, not the golden standards, but the best ways, if you want to see risk factors of frailty or identify potentially causal factors, of course, RCT is the standard way to go. This is, of course, always not possible or feasible. For example, if you would want to look at how physical exercise affects frailty, so then you would do an RCT on individuals who are frail or relatively frail, and see if physical exercise improves frailty. There have been some studies. The results so far have been a bit mixed. Some positive reports and some null findings too. And then there are these beautiful methods, statistical methods that you can use to infer causality, such as Mendelian randomization. Again, I'm not sure how familiar you are with that. But in Mendelian randomization, one uses genetic variants as instrumental variables. For example, if you would want to look at vitamin D levels and frailty, whether vitamin D levels are, let's say, protective for frailty, then one would use genetic variants from large scale GWASs, genome-wide association studies, that have shown the associations of these genetic variants with vitamin D levels. And then you go on testing the association between these vitamin D genetic variants with frailty in a series of analysis that I will not go into. And in the end, you will get the result whether there is support for a causal relationship. But then, of course, a lot of assumptions and things like that need to be met. And then it's called randomization because the allele that you test, in this case vitamin D alleles, they are randomized at conception. So this is creating, if done well, if designed properly, then Mendelian randomization can create approaches that are really similar to these randomized settings. And then in longitudinal studies that we have been doing and are about to do on frailty, if you have over a long period of time, let's say for 10 years or more, hopefully, you have measurements, repeated measurements in frailty, and then the risk factors. For example, we can say physical exercise. If you have repeated measurements in both, in these specialized models, you can assess their temporal dynamics. For example, whether changes in other measure, let's say physical activity, are associated with subsequent changes in frailty, or the other way around. So this is not the method to assess causality anyway, it's just telling about temporal dynamics between these two variables, frailty and physical activity, in which way the changes take place. And then, of course, genetic predisposition. One can do molecular and quantitative genetic approaches. I will come into those in a bit as well to find of course, the underlying genetics, and then genetics usually also pinpoint the underlying pathogenic mechanisms. And then nowadays, when one does GWASs, so these genome-wide association studies, there are really nice methods to derive genetic correlations. So for example, if you have genetics of frailty looked in at this GWAS, we can see using different types of methods what conditions are genetically related to frailty. So kind of genetic relatives to frailty. And then if one wants to look frailty itself as a risk factor for other late life or aging outcomes, then RCTs, yes, again, they can work, but Mendelian randomization is unlikely to work for frailty because frailty is so pleiotropic, as we've learned in our own studies, that the directional pleiotropy assumptions in Mendelian randomization, they will be violated, at least to my best knowledge. So that's unlikely to work. But instead, we can use this co-twin control design, which is a really nice way of not as powerful or not as good in not able to derive similar causal inferences as Mendelian randomization, but something similar. And I will tell you about that in a bit. But first, because it's related to twin or sibling analysis, I will first go through this quantitative genetics method called the twin modeling, sometimes called the classical twin design. And in this twin design, one can play with monozygotic versus dizygotic twin pairs. So we look at correlations within pairs. So correlating twin 1 in one pair to twin 2 in the same pair, in monozygotic twins separately, and then the same in dizygotic twins. I will also explain that in detail in a while. But here from twin modeling, the idea is to get out how much of variation on any trait, let's say frailty, is due to additive genetic influences, which we call A, dominant or non-additive genetic influences, also including gene-gene interactions, that is called D, and then shared or common environmental influences, including early life exposures, everything that is shared within a pair. So for the both members of the pair, they are the same. The magnitude of these effects can be teased out in twin modeling. And then there is this E component, which is unshared person-specific environmental factors and exposures. And this also includes measurement error, goes into this E component. So how it is actually done, total variance of any trait is the sum of these components that I just showed. And then if we look at, again, we take MZ correlation, and here I mean within pair correlation, so correlating member 1 in one pair to member 2 in the same pair. So additive genetic effects are perfectly correlated because MZ twins share 100% of their genome, so it's similar. And also they share these components to 100%. Whereas in dizygotic twins, because they originate from two zygotes, here the additive genetic correlation is 0.5. So they, on average, they share 50% of their segregating genes. Common environment is the same, but then this dominant gene-gene interaction effects, they are estimated to be roughly one-fourth the same in one pair. So these are the correlations, or this is the basis that we play with. And then when we pull out these correlation within pair correlations, we can see straight away very easily before even going into the actual modeling step that the extent to which the MZ twin pairs resemble each other more relative to DC pairs gives information of the relative influence on whether there's genetics or environmental factors affecting the trait. So for example, if we a typical most clinical or let's say meaningful phenotypes have an AE model that is explaining the variation in that trait, and that means that there's additive genetics and then this person-specific environmental component. That is the most, I'd say, maybe most common form. And for that to happen, we usually see that the MZ within a pair MZ correlation is well, let's say this way, the DC correlation is half of the MZ correlation. So if we see an MZ correlation 0.5, we would see a DC correlation 0.25, that would pretty much straight away indicate that this is an AE kind of trait. So additive genetics and unique environmental influences. And usually, let's say we see a 0.5 correlation in MZ twins. In the modeling, it's usually so that when we adjust for covariates and things like that, the heritability of that trait will be 0.45 or max the 0.5. So the MZ correlation gives you always the maximum estimate of heritability of any given trait. And then in this modeling, we of course determine whether it's the ADE model versus ACE model, because this cannot be, C and D cannot be modeled in the same model. So this is relatively, I don't know, technical way of assigning it. Sometimes it's even not clear, but it sometimes gives very nice cues in new traits, that how are they influenced by genetics or environmental factors for that matter. And then again, if we look back at the hypothetical MZ-DC correlations, these C and D work in opposite directions. So that D reduces the DC correlation relative to MC correlation. So let's say if DC correlation is greater or much less than half of the MZ correlation, so much smaller, that would indicate the presence of D. Whereas if there is C, so common environmental early life influences coming into play for that trait, then the MZ and DC correlations tend to be more similar with each other. And the whole point of the twin modeling is to determine which combination best matches with the trait. So there's a couple of words of twin modeling, and there is this co-twin control design. As I mentioned, it can be used to derive some sort of similar inferences as in Mendelian randomization, not exactly as strong, but still, if an exposure would be truly a cause of any outcome, if we do this co-twin control modeling, we would expect to find that the association that you find in any population level, say you have a normal cohort sample and you discover an effect size, let's say 0.5 again, then when you look the analysis in twin pairs, MZs and DCs separately, you would have to find that the effect size remains when you do the twin pair analysis, in which you control, in DCs, you control half of genetic influences and all shared environmental influences. So again, the kind of C component. But then if you find that your population level effect is attenuated a bit in DCs and even further in MZs, so if the MZ estimate attenuates relative to DC, then you can conclude, okay, there are genetic effects explaining this association. I will show a real life example from one of our own studies in a bit. And I will also illustrate how it actually works with this example, how it looks. So in a scenario where you have this IL individual level estimate, this is like a normal cohort estimate. If you find the effect size of this big, and then you do the within pair analysis in DCs, where you adjust for shared environmental effects fully and half of genetic effects, and in MZs when you fully account for genetics and environment. And if the association or effect size, we're not talking about p-values or anything here, because these sample sizes go usually down so much. So if the effect size remains the same, then you can say that, okay, yeah, this is consistent with the causal hypothesis. This is giving support to a causal hypothesis. It's not the proof of that, but it's giving support. This is, of course, very hypothetical and unlikely scenario in real life. One would have to convince the readers very hard if this kind of association would take place. And this scenario is again in your population sample. If you observe an effect size like this, and it goes down a bit in DZs and totally down in MZs. So this is a scenario where the association is entirely due to confounding. And by confounding, I mean genetic effects and the shared environmental effects. And this doesn't provide any support for a causal effect. However, this is actually quite common for many traits or for many association studies, where the effect is actually weakened and the effect size goes a bit, but it still stays. So it's still, let's say, greater than 1. So we can say that it's partially mediated by genetics or shared environmental effects, but there is still something that partly potentially causal. Right, and then let's see. So I'm going to present this study where we did latent growth curve model, where we used time varying risk factors for frailty. And we found, these are the population trajectories. We first found that there is this, these are actually back transformed values from square root values. So that's why they look a bit curvy. So we find that there's sort of an inflection point at the age of 65 in frailty trajectories measured using the frailty index. And here we divided that by weight categories, because here we found that in addition to these risk factors, deviations from normal weight, be it under or overweight, were associated with, let's say, worse frailty, so higher frailty. And we found that that underweight was associated with higher increase in frailty up until age of 65, and also continued being so. But then we observed something of that relates to a concept called obesity paradox, that towards the oldest ages, it was actually not that bad to have a bit of extra weight, because these who were normal weight accelerated more in their frailty towards the oldest ages than those who had overweight or were even obese. Of course, we were low on sample size at this age interval, and we were now replicating this finding in a study where we have more of these very old individuals. And maybe I'll see where we are time-wise. Maybe I'll go this through quickly. This is one of the studies on the risk factors of frailty. But then we did the genetic genome-wide association analysis in the UK Biobank for frailty index to look at genetics of frailty. This paper is now in second revision, hopefully submitted soon. So here we found this peak in the HLA region, and we found a bit less than 6,000 genetic variants associated with frailty. And of these variants, 26 were independent signals. And when we looked at this, so this color coding indicates that these red variants, so they are previously identified for some other traits, any other traits. We looked through the GWAS catalogs and all curated databases for previous associations. But these gray ones, so they are novel, or let's say unique to frailty. So they haven't been identified for any other trait before. And in this study, we also estimated so-called SNP-based heritability of frailty to be 14%. It's rather low, but that's often the case in SNP-based heritabilities. I will show you an estimate from twin modeling in a bit. But anyhow, then when we did a bunch of pathway analysis and predicted which cell type is more contributing the most to this variation and these kind of simulations, we found that there's pathway enrichment in these synaptic pathways, which is kind of not what we expected. So that frailty is somewhat of maybe neurological disorder too. And then we looked at the genetic risk score of frailty with its relatedness to other known phenotypes, where we had the genetic risk scores available. And we saw that frailty is most strongly related to genetics of educational attainment. So higher education is leading to lower frailty, put it that way. And then high BMI, having inflammatory bowel disease, waist-to-hip ratio, and C-reactive protein, having, let's say, high levels on these were associated with high levels of frailty too. And then we also did a Mendelian randomization in the context of this study, and we found evidence for a causal relationship between education and frailty. So higher education is protective of frailty, using all these different Mendelian randomization methods. And then we have some ongoing and upcoming Mendelian randomization analysis. We just finished, which is in revision, paper on vitamin D and frailty. We found no protective association, which is an important message because in some observational papers, I often see there is an association. Low vitamin D level is associated with higher frailty. And based on those rather, well, not weak, but maybe not so capable of inferring causality, those kind of reports, they sometimes write vitamin D supplementation is recommended for individuals who are frail. But that is actually not the case, because there's a whole host of reverse causality, which we can tackle in Mendelian randomization analysis. So it would be so that it's rather frailty itself that is causing the low levels of vitamin D, rather than the other way around. Because when you're frail, you move less, you go less outside the sunlight, you may be not able to follow healthy diets and things like that. So this is an important piece of message. But then again, for sarcopenia is muscle weakness and closely related to frailty. And there is some evidence on RCTs that vitamin D actually improves that condition. So that's why these more detailed studies are needed to disentangle whether some let's say protective agent like vitamin D, whether it is actually protective only for specific conditions and not so much for other. Right, and then we did these MR studies too. We see some evidence, some support for causal evidence. not so strong. And then we also did physical exercise and frailty, but here the Mendelian randomization assumptions were violated, so we couldn't conduct the study. And then this is our own study of the twin modeling on the genetics of frailty. And in this study, this is also now submitted, so we found that the heritability of frailty, roughly based on these various components, is 50% and drops to 45 when adjusting for confounders. And we also found this well, rather interesting finding that genetics play a greater role for frailty in women than in men. Then we have looked at frailty, whether it predicts late life outcomes such as mortality, and this is basically nothing new. There are many papers like this, but what we did in this study, this is relatively new, so we not only looked at all-cause mortality, but some cause-specific mortality outcomes too. And in this study, we used this co-twin control design to see whether we can find support for a causal hypothesis. And here in turquoise, we have the cohort effect size, so population level effect size. And here in pink, we have the DZ, and in purple, the MZ effect size. And we can see here that for all-cause mortality, in cardiovascular mortality and mortality due to respiratory causes, both in men and women, we actually do find support for causal effect, but nothing for cancer mortality. And this is a bit surprising too. And then we also looked at these fractions of or proportion of mortality or deaths that can be explained, that are attributable to frailty. So we calculated that on theoretically how many deaths could we hypothetically prevent by reducing the levels of frailty in the population. And these are presented again for the same causes of deaths that I showed in the previous slide. For cancer, it's of course null because there is no true association. But for all-cause CVD and respiratory mortality, we see that from midlife onwards, somewhere between 20 and 30% of all deaths could be theoretically avoided by reducing the population levels of frailty. And these effects go down towards the oldest ages, and this was also a bit surprising. And in the context of this paper, we also looked at whether frailty poses a greater risk, relatively greater risk at midlife than in old age. And we found that yes, relatively greater risk is found in midlife. So of course, in older age, the absolute risk of mortality is always greater. So there is no way around it. But then let's say that people who is 55 years of age has a level of frailty index 0.2, which is already quite high, and the same 0.2 level of frailty is found in an individual who is 80. So it actually is more detrimental being frail for this younger person than for the older person, for some reasons that we cannot currently explain. Maybe because in old age you die, you can die to so many causes and there are so many players and also stochastic events. But at younger ages when it's not so common to die, then if you are frail, then or frailer, I should say, so then it's a greater risk factor for you in relative terms. And then, so sometimes frailty is also called a marker of biological age or fitness, or how fit for your age you are. So we looked at frailty and other markers of biological age. Here we had telomere length. We had these DNA methylation ages or epigenetic clocks from Horvath, Hannum, and this is Levine pheno-age clock and Grimm age clock. And then we had this physiological age according to a Clemera-Dubal model. Maybe not familiar to you, but this is measuring your physiology, how old you are in terms of physiology. And then we had also cognitive capabilities, and then a measure called functional aging index and frailty index. This functional aging index is pretty close to what is the frailty phenotype of according to Fried. definition. Well, I'm not going into those in detail, but what we found that we first correlated all these markers with each other, and we found that seemingly there is a strong correlation between these all different markers of biological age. But when we adjusted these correlations for age, so used residuals of these measures, and one way to put it, we found that there actually wasn't that much left anymore. And we concluded that all these measures of biological age, they reflect different aspects of biological aging, or they capture different aspects of that with aging. And then we looked at the mortality and all these biological aging measures. How do they predict mortality? We first tested them one by one. So each measure was entered to the model alone, only adjusted for some age and sex and things like the covariates like that. So all except telomere length and this physiological age were associated with mortality when assessed separately. But then when we put all these markers into the same model, so kind of adjusting mutually for this, we found that the Horvath clock and the Grimm age, the other epigenetic clock, and the frailty index, only those remained independently associated with mortality. So this is telling us that well, it's not totally right or wrong to say that these are the strongest predictors of mortality. Yes, they are independent, and they also explain the same part of the mortality risk than these other measures, but do it better in one sense, and that's why these remain independent and most decisive for mortality, I would say. And then maybe I'll skip this one too. And this one. Right. So then now we're facing the COVID pandemic, and in the face of this, we of course have looked at frailty and the COVID mortality and comorbidity as well on the side. So in this study, just came out in summer, we looked in hospitalized geriatric patients. We used this clinical frailty scale and Charleston comorbidity index, that is a cumulative measure of, or let's say composite measure of your comorbidities. So we found that both of them predicted or were associated with in-hospital mortality due to COVID. And also adding these, this Charleston comorbidity index and the clinical frailty scale, adding that to the model where we had only age and sex of the patients. So we could obtain largely improved predictions using that. And then now, just recently, we did the same in community samples. We used the UK Biobank again with the linkage to the UK death records. And we found that when we add, we again have a model where we have only sex and age. And when we add a frailty measure, this particular measure, this hospital frailty risk score, or Charleston comorbidity index, adding either one of them actually gives really good, really accurate predictions on mortality. And we don't need any of these, let's say, demographic variables such as socioeconomic status or smoking or income or deprivation, social deprivation. We also tested. So fairly good predictions could be obtained using either the frailty measure or comorbidity index. And this is how it looked. So we not only tested the hospital frailty risk score that turned out to be the best, but we also tested the frailty phenotype and the frailty index. We found that, so these are the areas under the curve, which is, in short terms, it's indicating how accurate predictions you can get with your model with these parameters. So in model 1, this yellow line here, we have only age and sex, and we don't get very good predictions. So usually the 0.8 is required for to meet the threshold of good prediction or clinically useful prediction. And we didn't get any much better predictions adding the frailty phenotype or the frailty index to the model. So the area under the curve remained roughly the same as we see here. But then when we added either the hospital frailty risk score or the Charleston comorbidity index, the predictions became much more better or accurate. And here we see those curves. And then this last example of our own example using the co-twin control design for frailty and dementia. So we did a series of models. We had a relatively large sample of twins, and we divided that into full sample of individuals from their midlife to old age, and then a cognitive sample, which is individuals older than 65, because we call it cognitive sample because they had the cognitive assessment score available. So only for that. And we found that even if we did the multivariate Cox model where we adjusted for any other risk factors that we knew for dementia, frailty remained predictive of dementia. Then we had the subsamples of these both samples that had the genotype data, and we adjusted for ApoE, which is the most common single genetic variant known for increasing the risk of Alzheimer's disease. We adjusted for that. We still see an effect. Then we did a competing risk model where we adjusted for the competing risk of death, because that's another possible outcome. People can also die with or without dementia. And if they die without dementia, then this is creating a scenario of competing risk, and we need to take that into account in the modeling. Still, we see an effect. And then when we went into this co-twin control design within pair analysis that I explained a couple of slides ago, so we see that, okay, yeah, we see still an effect. The effect size pretty much remains in the DC twins. So let's say that familial effects are unlikely to account for this association. But when we go to the analysis of monozygotic twins, we see that this association is attenuated to null. Still some effect size remaining here, but in this older population, but here in this younger population, it's somewhat going to the null. Some remaining, but some not. So then we concluded that this is partially explained by genetic factors. But this is only half of the truth, and now we're running more detailed analysis because we actually show that this is an age-dependent or age-varying association. So the older you get, the less important the familial and genetic effects become. And we actually see a support for causal effect in the oldest ages. So when we look only at individuals who are 75 or older, so there we can actually see that the frailty has an effect that is consistent with the causal hypothesis. But when we look at the younger individuals, when we take a sample that is restricted to those who are younger than 65, then this is completely attenuated. So that's what we are doing right now. And with that, I'd like to thank the whole of our group, all the collaborators and the funders too. And with that, I'm ready to wrap up and take some questions.